So in this video we will discuss about MHC and some general characteristics of MHC. So the terms includes when we study major histocompatibility complex. Remember one thing, histocompatibility means compatible with means histo means tissue. So compatible with own tissue or cell, so it is called is majorly present, and if you classify it, it will be class 1, class 2 and class 3. So class 1 is found in nearly all nucleated cell, and class 2 is generally found, and remember one thing, they are all glycoproteins. Glyco means these are carbohydrate unit, and protein means polypeptide unit. So class 2 MHC is only present in macrophages, dendritic cells and B cells. And class 3 is a very different, and this gene, class 3 MHC gene, encodes some components of complement pathway. Component of complement pathway. So now there is some terms which are related with major histocompatibility complex. These are polymorphic genes, and they are polygenic also. So at first discuss the polymorphic. This is an easy term, that there is multiple form of same gene, which is actually called alleles. That means multiple alleles are present. Okay, and remember one thing, there is a interesting fact. Suppose this is the chromosome and there is some locus, there is multiple MHC gene are possible, and they are very closely linked. That means if you zoom it out, they are very close. This is the first gene, a bit gap, and the second gene, bit gap, very close to each other. So we know when we study recombination, if the two genes are very close, they are called linked gene. So linked gene, the more they close, the less their probability to be participate in the recombination. Generally they have recombination frequency of 0.5%. That means the difference between each gene is 0.5 map unit. So there is a statement, just carefully listen about it. Most individuals inherit the alleles encoded by this closely linked loci as two sets, one from paternal and one from maternal. So they are getting two sets. So this set of allele, each containing, suppose paternal contain 3 set of MHC gene, 3 set, and mother maternally contain around 2, 3, whatever you count. So the set of alleles is called a haplotype. Very important term, haplotype. So when you study standard books, you will see some terms called syngenic mouse and congenic mouse. So I'll clear these terms at first. In first lecture we will clear some terms. Syngenic means, suppose syngenic some mouse you consider. Syngenic mouse means two mouse. Suppose this is a first mouse, this is a second mouse. So they are actually inbred, and syngenic means they are identical. They have chromosomes. They are identical in all loci. Okay. genetically identical. You can say they are genetically identical. But what about, that means if a mouse is, or especially two strains, first and second strain are syngenic, that means they have identical or all genetic loci, they are actually identical. And finally there comes a term congenic. Congenic means they are also identical in all loci except a single loci, which is very useful in immunological uh, attached with molecular level experiments. So congenic mouse means they have all loci. Suppose this is strain 1 and this is strain 2 chromosome, they have thousands of loci, they are all equal in all, identical in loci, but except just a single locus they are different. For a single locus. This is called congenic. Syngenic are totally identical. So these are the basic terms for first lecture, and the other terms which are related to major histocompatibility complex and basic idea will be discussed in the next video. Thank you.